the mold? Well, the yeast phase on the right, notice it's a very large yeast cell, and the point of attachment for the bud and the mother cell is quite broad. That's classic. The mold phase on the left that we see growing in the laboratory is a single conidiophore with a bud coming off of it. It looks almost like a balloon on the end of a string or a lollipop on the end of a stick. So this is clearly a dimorphic mold. And this is Blastomyces dermatitidis. The next slide, name the genus and species and define the category of mold. And then how does a person get infected with this mold? Well, you can see that there's a yeast phase on the right and a mold phase on the left, so we know it's a dimorphic mold. The genus and species, did you say Sporothrix schenkii? If so, you are correct. The yeast cells are, I'm searching for the word here, I'm sorry. The yeast cells are pleomorphic. Some are small, some are elongated, some are peg-shaped, some are attached with a very thin line of attachment. So this is a classic yeast phase for Sporothrix schenkii. And the mold phase is a very delicate, thin conidiophore with microconidia attached at the end. How does a person get infected with Sporothrix schenkii? The other dimorphic molds we've talked about are respiratory infections. So the spores are breathed into the lungs. Sporothrix schenkii is not a respiratory pathogen. It's a dimorphic mold, but it is not systemic, and it's introduced through the skin. A classic example is the prick of a rose bush. So it gets into the skin and then the lymphatics. As you can see, dimorphic molds are important, and students need to know those. They're always considered pathogens. There are two more dimorphic molds that we're not going to talk about, and that's Paracoccidioides brasiliensis and Penicillium marneffii. Okay, name the genus, and what category of mold is this? Both of these images are the same genus, and these are macroconidia. So those on the right, if you notice, are thin-walled macroconidia, and the ends are rounded. Compare that with the macroconidia produced by the mold on the left. They're spindle-shaped, and notice that they are rough-walled, and that the end of it comes into a nice point. So the genus name is Microsporum. And it just so happens that the one on the right is Microsporum gypsum, and the one on the left is Microsporum canis. And do you know the category of mold? Well, Microsporum is a dermatophyte. This next slide is isolated from the hair. Well, the slide isn't isolated from the hair, but it is a picture of a mold that was isolated from a hair sample. What is the genus? And can you name the species? If we look at this, we see all kinds of microconidia. We see those that are nice and round, others that are teardrop. We see conidia that are peg-shaped. So they really vary in size and shape. And this is a trichophyton, probably a tonsorians, because of the variability of the microconidia. This is a dermatophyte as well. Name the genus and what the category of mold it belongs to. What are you seeing here? Well, we're seeing long conidiophores, and on one end is a big sac that has broken and the spores are spewing out. On the other end of the conidiophore is a root-like structure or a rhizoid. So, is this a zygomycete? Yes, it is. That's the category of mold. Zygomycetes have these broad hyphae that are usually non-septate. And they have reproduction that occurs in these sac-like structures that are called sporangium. So what is the identification of this mold? We know it's a zygomycete. If you said mucor, that's incorrect, because mucor doesn't have any of those rhizoid structures, or they're very weak looking. If you said it's Absidia, which is now called Lichthymia, that's incorrect, because the rhizoids don't appear directly below the sporangium and the conidiophore. And if you said it was rhizomucor, that has branching conidiophores. So the correct identification for this is Rhizopus. Remember that zygomycetes are rapid growers, and they really have a lot of aerial mycelium. Name the possible organism. So look at this image carefully. We see some red staining filamentous structures. Parts of it even look beaded. This is really a trick question. This is not a fungus. It's not a mold or a yeast. It is an acid-fast stain, and the identification of the organism is nocardia staining filamentous structures. Parts of it even look beaded. This is really a trick question. This is not a fungus. It's not a mold or a yeast. It is an acid-fast stain, and the identification of the organism is nocardia. So remember that it can be filamentous just like a mold can be, with the beading in some areas. Trick question for you. The images in this mycology review were from the public domain, that with the CDC, or with permission from University of Texas and American Society for Clinical Pathology, and I thank them. And I thank you for your attention. Okay.
So for the next meeting, would you want to, tell me when you want that meeting or recording.
Well, I can do it before